So good afternoon, everyone. Thank you everyone for joining us this Tuesday. And my name is Audrey, and I am the event coordinator as well as the secretary of the Student Advisory Council of CAP. And I'm joined by Ireneo, the graduate chair. And today we commence the physics hour series with Dr. Claudiu Gradinaru from the University of Toronto. So thank you again for accepting our invitation. And before we begin, please feel free to put your question in the chat and we will have a Q&A session at the end. Also, let's keep this respectful. CAP has a code of conduct to be followed. Other than that, just enjoy this hour. And once again, thank you for coming. And with that, I will now turn it over to Dr. Gradinaru.
All right. Thank you, Audrey, for the introduction. Welcome, everyone. It's my real pleasure to talk a little bit about what I'm doing in the darkness of my lab here at the University of Toronto. I'm on the Mississauga campus, the western campus of U of T. I'm a physics professor here for almost 20 years, and I'm a biophysicist. You see from my title, less is more. What my lab is basically doing, we're looking at biological molecules one at a time, and using all kind of photonics gadgets and tools to really go beyond averages. So, okay, let me see. First, let me motivate my work. If I should do it maybe from the get-go. Why go this level, single molecule? Why aren't averages sufficient? Well, first of all, the object of our study, biology or biological molecules, are often appear in multiple states simultaneously. So we're dealing with heterogeneous mixtures, talking about folded and unfolded proteins, aggregated, unaggregated, bound, unbound, all kind of different states that coexist. And I don't probably have to convince this audience that averages can be misleading. For instance, you have, maybe I should go to a laser pointer. Pointer options. Yes. Yeah. So for instance, if you have a probability distribution of states, two states, and so you measure this average, I don't need to convince you this average doesn't correspond to any physical microstate. So that's why it's much better to get access to the full distribution of states. And the other part is often when we measure averages, not only the averages can be misleading, but we can entirely miss minor populations. And in biology or in life sciences, these aberrations, these minor populations often have pathological implications. So that's why it's important to try to capture as much information about the entire distribution of states that a biological system presents. So how can we detect single molecule? I use fluorescence. So the two major things. Basically, it's a signal to noise problem. You either increase the signal or reduce the background, and at the same time, you must make sure that you only look at one molecule at a time. So how do we do this? If you take a laser beam and you focus it with a diffraction limited spot to using a nice objective, you basically can focus the light on a volume that is on the order of a cubic micron, which translates in 10 to the minus 15 liters or a femtoliter. And you can calculate easily that how many water molecules are in there. There are about 10 to 100 billion water molecules in that volume. And you have one molecule of interest. So imagine that your molecule has to produce a signal to overcome such a huge amount of water. And you may say water molecules is inert, but it's not entirely true. Solvents contain impurities. So these kind of things put some strict restrictions on how we prepare our samples. Well, you can increase the signal, and the arguments I'm giving here is some back of the envelope calculations. What is brightness in principle? It's how many photons you absorb multiplied with how many photons, percentages of photons emitted. So if I absorb 100 photons and I emit them all, then I have a very good brightness. But and the signal is typically brightness multiplied with the number of molecules in your detection volume. Why is water not your friend in this case? Water produces background signal via Raman scattering. So take a water molecule. The brightness of a water molecule in some units is 10 to the minus 29, but you have many of them. So there's the signal that comes from water in your detection volume is it's on the order of 10 to the minus 18, say arbitrary units. If you look at a protein, the building block that has some optical activity fluorescence is called tryptophan. The tryptophan has a much higher brightness. You see more than 10 orders of magnitude, but you only have one of it. And this tryptophan gives about the same signal as all your water molecules. So this is not a good idea to use the natural occurring molecule in your proteins as an emitter, as a marker. So that's why what we do, we use chemical, so dyes or fluorophores of molecules that have extended pi systems, conjugated bonds that can emit much more. So their brightness is, you see, is 100 times larger than that of a tryptophan. So basically can go to about 100 to 1 signal to noise when you compare the signal from fluorescent 10 to the minus 16 with the background from water. The other stuff that you need to make sure that occupancy, you look at one molecule at a time in your detection volume. So as you know, in a solvent, if you have a box, you have a Poisson statistic. So the probability of having, say, a K molecule giving an average M is given by your Poisson statistics. Basically, the condition that we need to ask is this, that the probability of having 2 or more molecules should be less than 1% of probability of having one molecule. And when you impose this condition on the Poisson statistics, you have that on average, you have 0.02 molecules in your detection volume. So most of the time, you have nothing. Only very briefly, occasionally, you get a molecule passing through. So when you take this average number of molecules and you divide it by the volume, say a femtoliter, you get concentrations of samples that are 50 picomolar, so about 100 million molecules in a drop, very dilute samples, right? So you need to ensure that you operate in this regime, otherwise you'll have you have more than one molecule at a time to study. So basically when you take all these conditions, you put bright labels on your molecule of interest, you dilute the samples, you get this kind of spikes, bursts, and you don't have negative photons here. These are like two labels at the same time. So you get bursts of photons that last about a millisecond and contain 50 to 100 photons on average. And that's where our business is, analyzing the information that comes from this intensity bursts of photons that may look like gamma ray bursts, but in this particular case are just fluorescence bursts coming from our proteins diffusing through a tiny volume. So what information can you learn from if you take the time, make all the effort to go to single molecule observation, single molecule fluorescence? So this is maybe a busy slide, but what we in this business, what we do, imagine that your signal, the signal is constituted of photons, and in our case, we detect each photon one by one, each at a time, and we put labels on it. What color? When it arrives with respect to a laser pulse, you have a train of laser pulses. What is the absolute arrival time? So we have a list of photons, and then we can do all kinds of different analysis on different time scales that inform us on various chemical or biological time scales of our molecules of interest. And I also have here 2 different modes of detecting this single molecule fluorescence. I'll talk about them both and with applications to proteins that I study in my lab. One is a confocal mode, confocal microscope, and the other one is a total internal reflection. Just to give you an idea what sort of analysis one can do with this photon information, look at arrival time with respect to the laser pulse, and you get so-called fluorescence lifetime. You get the excited state lifetime, which depends on the environment, depends on various things. You can get polarization information, and isotropy tells you how rigid or how flexible the environment of your probe is. You can also measure autocorrelation. So this looks at, unfortunately, I don't have time today to talk about this technique, but we do have a setup, an instrument we built that looks at intensity fluctuation. And this tells you something about quenching, about diffusion, so you can learn a lot by doing this type of analysis directly in time domain. One thing that I'm going to insist today is this measurement thread, which will allow you to measure molecular distance on the nanometer timescale. Enough of this. Let me show you a picture of how my setup looked about 2 years ago. Now it's a little bit different. So it's really like an optics, photonics lab. We only buy the lasers and the detectors. The rest, our microscopes look like boxes. I don't have a picture from inside the box there, but it's basically stages and mirrors and mounts and crystals and all that stuff. Now, the thing doesn't look like this anymore because we acquired a very cool laser about 3 years ago, a super continuum. It's a white laser. It emits pulses about 10 picosecond in duration every 25 nanoseconds. And look at its spectrum. It's really going from 450 to 2500 nanometers. So with this white output, you can basically excite any label you wish. You can excite blue labels, red labels, infrared labels. You can do multi-photon excitation. So it is quite a versatile tool to have. Not only that, but you can choose up to 8 colors simultaneously. We're modulating, alternancing of pulses. You can play all kinds of games in terms of excitation and or detection modulation of your signals. So let me see. Pointer. Oh, this doesn't. Sorry about this. There is a thing. Yeah. Okay. So what you see here, I mean, I'm not going to go through the details of this thing, but imagine that in the confocal microscope, your molecules, this is a movie, an actual movie of the burst coming from our machine that was measured by a grad student a few days ago. This is a piece of DNA where you see bursts of blue and green emitting photons. So we have a molecule that has 2 types of labels on it, and we see this burst coming on in real life. And these are not random bursts. These are fluorescence bursts coming from molecules passing through our detection volume, right? As I explained to you, you may see only the intensity here, but behind this, there is information about arrival time of the photons, the color, the polarization, and that allows us to do all this multidimensional analysis on our signal. So one very popular technique is this one called first-order resonance energy transfer, which basically it's a dipole-dipole transfer between, you excite say a green label, and you see signal coming both from your original label, green, and from another label that is placed at a certain distance from the green label. And if the red label is closer to green, you get increased output from the red upon excitation of green. But if the distance is larger, you primarily see green photons coming out. So this allows you to look at distances on the nanometer timescale and also fluctuations of distances on the nanosecond, microsecond timescale, and so on. So the cool part about this, it's a very sensitive to distance. It's 1/r⁶, pointing to the dipole coupling between the two dipoles. So before I show you some examples of the data, what do we do here is, again, I'm alluding again to this burst, the train of bursts that comes out. What do? We identify, we have some criteria to identify burst above the background. we count number of photons sorted by wavelength range, number of photons, and say, let's take this burst. I have, I don't know, maybe 60, 70 photons in this green burst and similar number of photons in the red burst. And then you take a ratio between red versus total, and that we call efficiency of energy transfer, right? If you have 0 over ND, that's a 0 efficiency, that would be here. If ND is much smaller, then you have close to 1, which is here. Anything in between, you get intermediate Fret efficiency, right? And basically allows you to resolve multiple populations depending on how sensitive your probes are and or how much conformational state your protein has. So the first system I want to tell you about is a so-called the dark matter of biology called intrinsically disordered proteins. What are they there? Whenever you think of proteins and crystal structure of proteins and electron microscopy, you think of kind of rather poses, structures, static pictures of these proteins. Now, there are a significant number of proteins in the genome and that are either fully disordered or contain stretches of disordered proteins. And this complicates life when you try to understand how they function, how they interact, how they bind to drugs. So these are not just a figment of people imagination, like what an interesting object, let me study it. No, it has implications for disease. See, they're implicated, and particularly in neurodegenerative diseases, in cancers. You see here the musculoskeletal diseases. So it's worthwhile because if you want to develop drugs against them, it's complicated. This lock-key mechanism of how classic drug development works cannot apply to this very dynamic system. So you need to come up with ways to characterize the conformational ensemble of these disordered proteins. So the system that we studied, we studied a number of them, but most recently we looked into this protein. It's called the 4E binding protein 2. What does it do? It's regulating translation. When I mean translation, is not the translation in the physical sense, it's the translation in biological sense, the conversion from mRNA to protein by the ribosome. So when it's bound to this, where am I? Here, yeah. When it's bound to this eukaryotic translation initiation factor, this protein is inhibited. It's arresting translation. When 4G replaces it, then the translation can proceed. So 4E-BP2, this protein that I'm studying, is an inhibitor of translation. And the way it's regulated binding versus unbinding is via phosphorylation. If it gets some phosphates on it, it detaches. It goes from a very tight binding to basically detaching and allowing 4G to bind and the translation to occur. So two of my grad students, what they did, they designed Fret experiments. So they looked at energy transfer, so distance information of between different segments in the chain all the way. This is 121 residues, so you can see probing various segments. This is N terminus, and this is all the way at the C terminus. What you see here, there's a lot of information, but what I'll give you an example. Of N terminus, the first 32 residues, when we measure this distribution in the free state, we get the histogram around the vertical red line. Now, when it's bound, it shifts to higher Fret efficiency, meaning that it gets the two probes get closer. Now, if we go to, and the same we see at the other terminal, but if we go somewhere in the middle, say between 32 and 91, it's exactly the opposite trend. If in the free state was here around 60%, 0.6 Fret, what it does in the bound state, it actually expands. So it goes to lower Fret efficiency. So majority of the distances, as you can see, shift towards lower Fret efficiency. Only at the end we have compaction. So this kind of gives you an idea of non-local compaction or expansion of the chain. What we find out, I'm not going to go to show other measurements here and modeling. We found out basically from this study that this protein, while it's bound, is not static. It's highly dynamic. So it kind of defies the rule of thermodynamics that you lose a lot of entropy when you get bound, and that's how you define tight binding. This protein can achieve tight binding by not paying such a high entropy penalty in the bound state and having a lot of dynamic interactions with the multiple states, like hugging the target rather than having a single binding site. So maybe I'm moving too fast. Maybe I should breathe a little bit because I should explain maybe a little bit more. But this is a larger part of my talk. I want to change gears to go to another technique that we use. This is total internal reflection microscopy, and most of you should know that this relies on evanescent field being localized close to interface between a denser medium like glass and less dense than like water. So this kind of gives you an idea. If you have a cell, this is a kind of a schematic of a cell. If you do wide field, so like a parallel beam, you have a depth of field of about a micron. If you do confocal imaging, it's about the wavelength of light, 500 nanometers. But TIRF, total internal reflection, is not 40, it's about 100 nanometers when you look at the penetration depth, how much the evanescent field, it's a non-propagating field, you don't see it in the far field. But if you have a sensor, if you have a fluorophore that is present within this 50, 100 nanometer area above the surface, it will be excited. And that's actually, in a nutshell, the idea behind using this technique to really inquire what's happening at, say, the bottom membrane of the cell. And that's what I'm going to show you. But first, a diagram of the setup. Again, we don't have that white laser setup here. We don't need pulse laser. The detectors are not avalanche photodiodes that detect single photons at a time. They are sensitive EMCCD cameras that capture frames, pictures from this bottom cell membrane. We have multiple lasers from blue all the way to red. And I have an acoust-optic tunable filter. A Raspberry Pi controls the timing and synchronization between various lasers and the acquisition. Multiple modalities can be deployed here. I'll give you, I think I'll show you examples from the first and third, but I'll briefly describe what they are. First of one is you basically can get images of single emitters, multiple single that are on the surface of your sample. Say whether in the cell or in vivo, you have pieces of DNA, and you get these spots, and you can look at the brightness of the spots, and you can look at how intensity changes in time. I'll show you an example of that in a minute. Now, you may choose to shine two lasers at a time, like say the green laser and the red laser. And what I showed you with this burst of photons, molecules passing through and only observing them for 1 millisecond. Now you can take the same protein and you can tether it to the surface, immobilize it, and then you will have a dual channel detection, a donor channel and an acceptor channel. So then you can look at FRET, the ratio of the two intensities over a much longer time scale. You'll see the resolution will be in the 10 millisecond range. That's how fast the frame is. But you can observe it for 10 seconds or even a minute sometimes if your fluorophore is photostable. So you see, you can do single molecule FRET, parallel measurements, multiple molecules, but for longer periods of time. You don't have the microsecond, millisecond resolution that you had on this. Last but not least, and very exciting, this you can do in live cells. You can track proteins one at a time or two different proteins, and I'll show you some cool examples of this type. So the first and third will be examples that I'll show you from this type of microscope. So you probably know these two guys. Just to give you a motivation, what system we're studying with this are, they're called G protein coupled receptors. So there are receptors, when you probably had your coffee, depending on which time zone you are. I just had mine about an hour ago, my afternoon coffee. But caffeine is an antagonist of one of the receptors we're studying in the lab, adenosine A2A receptor. It also acts on other receptor, GABA receptors, so ion channels. So this is a ligand, it's a drug that acts. What it does to this receptor, it inactivates it. So GPCRs, G protein coupled receptors, it's a ubiquitous class of transmembrane proteins. There are several families of them. They respond to a wide range of stimuli, from photons, lipids, peptides, proteins. You have small chemical, like caffeine, small drugs. They share something in common. They have like 7 transmembrane domains, various size of these disordered loops or C-termini. They are encoded of close to 1,000 genes in the human genome, and they are the primary target for FDA-approved drugs. About a third of the pharmaceutical drugs in circulation target GPCRs. How do they work? You see, you basically have the ligand, it binds, say this is your aspirin, your caffeine, it binds to your target receptor. The target receptor then gets activated. In the active state, it can engage its effector protein, which is called G protein. That's why this is a receptor that is coupled to the G protein coupled. Since it's the G protein, it's a trimeric protein. This one activates the G protein, and upon activation of the G protein, a nucleotide is being exchanged here, and this goes on to activate all kinds of other things. Adenosine cyclase to produce cyclic AMP, open ion channels, ATP synthase. So there's a cascade of signaling that all starts with this event, ligand binding. The different kind of ligands activating, this is agonist. Inactivating, this is called the inverse agonist. Partially activating, this is called partial agonist, or some that are doing nothing, they still bind, they're called neutral antagonists. They antagonists because they bind the receptor and the receptor is not available to be activated. So let me show you what we do with this. One of my previous grad students, Dennis Fernandez, if you look at this movie, this is the single molecule photobleaching, or the photobleach step counting. These dots, the brightness starts somewhere and then they dim in discrete steps, so they don't vanish at once. So if you would be to look at, say, one of these steps and see how the intensity changes as a function of time, you can quantify how many discrete steps each and every spot has between fully on and fully off state. And then basically that reports on the number of copies of the protein that exists in this particle. So these were experiments done on muscarinic M2 receptor, one type of GPCR. They were gently purified from cells and then put on the surface, and we count the photobleaching steps. What we learned from that primarily we dealt with complexes of tetramers, tetramer of M2 receptor coupled to a tetramer of G protein. So we did counting on both of these units. We tethered via G protein, we counted the receptor. We tethered the receptor and we counted the G protein. So a lot of painstaking work when Dennis and Robin from a collaborating lab in pharmacy went to do this. Now we can do much better acquisition than this from 10 years ago, but very cool technique to just quantify the not only oligomers, but also heteromeric mega complexes when you look at protein complexes. I'm not going to go further detail than this. Let me show you the coolest, latest applications from my lab. They are about looking at single proteins in live cells. And in order to do this, remember what I said to you in the beginning. To look at single molecules, yes, you need to have good signal to background. To have good signal to background, you also need to make sure that you only have a very bright molecule and not so many other molecules close by. So you need to control how your protein is expressed in live cells. So what we have here, Xiaohan, PhD student that is about to defend in my lab his thesis, we use this system that we can control the expression of GPCRs in live cells, and we label the GPCRs in situ, in live cells, with some very bright fluorescent probes from Janelia farms. And these are specific labeling. These labels, they don't go everywhere. The dark when they don't bind to their target, and when the labeling occurs, they go to a bright state. So it's a really good probe. So you need really to have friends that were to know your fluorescent probes. You cannot just take anything off the shelf. So what you see here is controlled expression from low single molecule level to high, close to physiological level. You can really get 90% of your cells in this preparation to look like this, 90% to look like that. So controlled uniform expression. For now, we did experiments on all three cases and with various modalities, super resolution, correlation spectroscopy, but I'll only talk about this case where you see in this movie, our idea was to track the diffusion of these individual particles in the bottom membrane of the cell. So what we see are objects moving around on the bottom membrane because we excite this using evanescent field, and we detect through the same objective the fluorescence coming from individual particles, whether some of them are monomeric, some of them are dimeric. So you can look at the intensity of each spot and you can create a distribution of intensities. But I'm not going to talk about the intensity for this. I'm going to talk about the diffusivity. So single particle tracking, which is the name of this technique, extracts positions from subsequent movement, builds tracks, and those tracks that are illustrated here allows you to really say something quantitative about the nano environment, to say something about the oligomerization, about the interaction with other proteins, about conformations. So how to do this is we use exquisite programs. These are developed by our collaborators of Polytechnique Montréal. It's François Simon. He developed a program called X-Track. Now the version is called ExaTrack. You take all these trajectories and you see some of them are colored in red, meaning that they diffuse further away, and some are in blue, meaning that they are more confined. And you look at all these trajectories and you say, I think I have a finite number of diffusion states that have a certain rates of transfer between these diffusion states. And that's, you try to find, basically using variational Bayesian analysis and hidden Markov models, what is the minimum number of states that you need to describe the data, not from a single cell, not even from a single dish, but from many cells and many dishes. So there is a repeatability problem here. It's not a single cell. You have a decent number of tracks here, but we have thousands and thousands of tracks from tens of molecules across maybe 10 to 20 dishes. Okay? So in the end, you get states that have diffusion constants, population fractions, and probability of transitioning between them. So that's, by the way, was Maria Shemeteva, a grad student in my group, did work on this with Shahan. So let me show you what diffusion states of GPCR. This is, I think it's a muscarinic M1 receptor. We looked at a number of receptors, but if you look, this is a snapshot of these dots, and at each and every dot, you can see diffusion tracks. Again, blue means that it's not moving too far. The further you go up, it kind of scales with displacement. So very large displacement, and then the yellow would be intermediate displacement. When you do analyze this, you get three states. Primarily in this case, you get 75% is Brownian motion, and this is the diffusion coefficient. You get another fraction that has about a factor of 2.5 slower diffusion, and we show via other tools that this is a confined state. So we're talking about these states. You see, it diffuses like cyan or yellow, diffuses, but it's confined. And then finally, we have a small 5, in some cases 10% fraction that is, we call this immobile. It hardly moves on the scale of our measurement, which in this case is about 60 seconds. Now, this was for the receptors. How do you think that the G proteins, which are inside the cell, the G proteins diffuse much faster because they are inside the membrane, inside the cell, and they're anchored in the inner leaflet of the membrane. So they basically diffuse much faster, and their state scheme is not linear. You see, it's triangular. All states are coupled, and there are significantly different populations of Brownian, confined, and look at the immobile, is quite different. So the way proteins are diffusing and partitioned in the cell, what our hypothesis was, is that it impacts the signaling output. It's not only how dynamic the conformations are, this receptor that people study in vitro, but in live cell, it depends where you are, how fast you diffuse, or whether you're confined or not. So in this particular case, what I wanted to point out, although this diffuses faster than the receptor, it's also more confined. And what we plot both for receptor and the G protein, all this allows you to come up with something called diffusion map. So at every point in the cell, we can color it when we weigh all the occurrences at that point, whether that membrane part is conducive to Brownian motion, like it's red, or if you see a lot of green, red, that veers towards confined or immobile. So this kind of gives you an idea how heterogeneous the membrane environment is for membrane proteins. In this particular case, G proteins, but to get diffusion maps also for receptors. The last part I want to show you about this is that we actually express at the same time the receptor and the G proteins, and we did dual color tracking. And I hope you can see in this plot that there are some areas where both the receptor and the G protein seem to diffuse in some confined areas. This is adenosine A2A, and it's cognate G protein. So we hypothesize that these are potential interaction regions where perhaps they interact, they activate, and they signal. So again, we inferred this based on diffusion alone. We don't yet have, we're working on to get activation sensors or signaling sensors. But the thing we did prove that these interaction regions are modulated by lipid rafts, so the lipid fluidity, cholesterol, they're modulated by cytoskeleton. So these domains exist in the cell because of the lipid composition and because the underlying cytoskeleton. And we think that they are the ones determining how the GPCR signal in vivo in this kind of signaling hotspots. And to drive this point further, I just want to show you a comparison between, I have here two different receptors, muscarinic M1, and it's cognate G protein. This is M1, this is G11, and this is A2A receptor, and this is GS. Different diffusion maps, same cell, different. So red is Brownian, and yellow, green is more confined. So at first sight, what do you see? That this one show more confinement on average than this one. So A2A looks more, and it's indeed true. And this, there is, if you look at the image analysis, there is cross correlation between the non-Brownian regions. That's what we plot here. The fraction of colocalization of non-Brownian diffusion in receptor and the G protein. So if you look here, this is the muscarinic M1. It has a lower fraction than the adenosine. So you see, look at this, it's about 30% colocalization with its G protein, whereas this one is 55, close to 60% for adenosine. So no ligand thrown at them. This has a higher co-confinement with its cognate G protein. I didn't show you here signaling assays, but guess what? This receptor doesn't signal almost at all without adding the ligand. So in the basal state has very little signaling. This one, adenosine, has significant basal signal. So when you add an antagonist, the activity goes down. So we think that there is a clear correlation between the core confinement and the basal signaling. That's what Chauhan's current paper that is under review formulates as a hypothesis that that's what drives this behavior. And to close this, why core confinement? When you confine to, I mean, the schematic that I showed you in the beginning, with the receptor being activated by a ligand, then binding to a G protein, then leading to a signaling cell, is fine if you think of a schematic like a biology textbook. But in real life, for two molecules, A and B, to meet, they need to find each other, they need to diffuse, and the way to increase this encounter rate is to enclose them in these confined areas. And that's where, when you solve the diffusion equation in a confined area, you find that the rate of encounter goes like this. And for the domain sizes that we measured, which are in the range of 150 nanometers, we see that we basically go by almost 2 orders of magnitude, speeding up the signaling, because that was the other question. Why do receptor signal so fast? I didn't show you data, but when you add the ligand within 100 to 200 milliseconds, which is close to this, you get a signal output. If you would believe in Brownian motion, then it would take quite a bit longer for the biological response to set in, 10 seconds or longer. So we do think that the nano-confinement, the nano-domains, the membrane organization is really important for signaling. It's not enough to just look at structures or conformational dynamics. Not that that is not important. So I was trying to stay within time, which I think I am. I think in summary, what I wanted to drive home is that single molecule techniques, single molecule fluorescence in particular in my lab, we have a wide range of techniques from FRET, which measures intra-intermolecular distance, correlation spectroscopy that looks at global sizes, local dynamics, and so on. Photobleaching that tells you copies of oligomers and mega complexes. Tracking that tells you diffusivity, trafficking, interactions in a lifestyle environment. So this is, when you combine all these toolbox, you can really get a super rich information of biological heterogeneity. You can look at complexes and dynamics on multiple timescales from nanosecond all the way to seconds. I showed you 2 examples of applications that my lab has been busy for nearly a decade. Intrinsically disordered proteins, and I didn't show you, but we don't only measure using single molecule techniques, we also try to integrate our data with other complementary data, whether it comes from NMR or X-ray scattering, to really come up with an ensemble description of IDPs. And GPCRs is cool because A, they're target for drugs, so they're medically relevant, but B, because they also have some interesting, funny behavior, which we think is not entirely, but partially driven by the membrane environment. Last but not least, I just want to mention that these techniques, I look at them as high throughput, high content that have a lot of potential to be applied and are already applied in omics applications, all kind of drug discovery and material science and so on. Last but not least, I want to thank the past members of my group, the PhD students, the current PhD students, many enthusiastic undergrad students, master students, postdocs, and fantastic collaborators all over the world. And I just want to leave you with this simple statement from George Fair. The difference between physics and biology is the following. One is difficult by simple, whereas the biology seems easy but is complex. So with that, thank you for your attention. I'm happy to take any questions you might have.
Thank you a lot, Professor Gradinaru. It was really fascinating to learn about how the ways you can analyze and observe proteins with optical system. So also thank you everyone for listening. and I'll open the Q&A session. So if you have any, you can either write it in directly in the chat or open up your mic to speak up and ask your questions. Yes, Benjamin.
Hi, so great to see you again, Claudio. It's now my turn to ask you questions about your talk. Um,
Go ahead, Ben. Now it's your time to take revenge. Go for it.
yes. So it's a really beautiful measurement how you can do those like single molecule, like time resolved fluorescence with the like the first kind of half you showed. I'm kind of wondering, I'm coming from this sort of from a perspective of like Raman spectroscopy. Do you ever get issues with like cosmic rays, or does like the time resolved like kind of gating, does that sort of solve that issue?
That's a good point. The cosmic rays, I think, worried me about 20 years ago when I was building my lab.
Hmm.
Because we also have these avalanche photodiodes and uh, the CCD camera. But to be honest with you, the main source comes from the electronic noise of this. That's the one that is. So that's why I didn't mention, but the avalanche photodiodes that we use for the burst setup have very low dye counts, like 50 counts per second. So
Mm.
um, these solvents, when we have to have spectroscopic grade, sometimes if you use the wrong pipette, you contaminate the buffer. So this we take really, you have to be the glass has to be super clean and all that. So there's a lot of you know secrets behind this to kind of ensure that your signal is really clean and it doesn't have to fight all this background. But I don't have other labs next to me like nuclear physics or other things that could potentially disturb. I have a bunch of chemists like inorganic chemists next door, and it's not really. I haven't seen yet perturbations due to nuclear radiation.
Yeah, that makes sense.
Yeah. Yeah, I'm not seeing the chat, so I don't know, Audrey, if there's anybody asking any.
Yes, Ireneo wrote certain question in the chat. You
Okay.
can.
Yes, there's a question from the YouTube chat. So it's
Yeah.
asking, I was wondering if the experiments are at body temperature and how temperature affects the experiments. Like, is there some type of trade-off using lower or higher temperature?
It's a good point. To be honest, it's done at 20 degrees Celsius, which is our temperature in the lab. It is true that for the live cell measurements, we probably should aim for 37 degrees. And we have a temperature control stage. We haven't used it lately. The problem with that is the following. You need to, it's not impossible, but priorities come and go. You need to maintain focus. You need to stay in the focal plane, and you need to correct for all these drifts that occur during your measurement. So if you try, for instance, to acquire single particle tracking measurements and your particles go out of focus and you think internalized, you may run into that problem. So we decided, the cells are happy for a couple of hours at 20 degrees Celsius rather than if we would want to do longer term measurement. Yes. We haven't seen thermodynamic changes over this small temperature range of 10 to 15 degrees, to be honest, for the proteins that we study. And in solution, to be honest with you, when I look at the single molecule threat or these bursts, the biggest perturbative factor is not the temperature, but the salt conditions, the pH. So the chemical parameters are much more important. The proteins are not so unstable that they get totally destabilized or stabilized by 10 or 15 degrees difference.
Great, thank you. I have some questions from myself. Okay, so the first one is that if you'd like to share, what are some future projects that would be available for prospective research assistants or graduate students in your group?
Yeah, I think I want to show you, I mean, I want to discuss a little bit is this. So I think you probably still see my screen. We see, oh yeah, this is, these things, what we do, I want to basically not only track to the receptor and its target as they move in the cell, but I want to be able to actually nail it, like to say, if two things had an encounter, is it a productive encounter or is it a unproductive encounter? And what do I mean by this? What I mean is, if the receptor, say, couples, even transiently with the G protein, do I have proof that it had a conformational change on the receptor? I want to be able to do single molecule thread while tracking these two proteins, which is a single molecule thread between the receptor and the G protein that only captures both the complex formation and the conformational change that signals, yes, you activated the G protein. It's a tricky process because to track, I can put a relatively big tag on outside here, the N-terminus. For the G protein, it's inside, it's a little bit trickier. If I want to do thread, I need to be minimally perturbative. I need to put very small probes to be able to capture this distance, just like I did for this other protein. That's it. I have students that are working on labeling strategies, but we also need to develop data acquisition analysis strategies. So we're starting to look into that, which is it's a fairly challenging but high reward, high risk program to go and actually get not diffusion maps, but to get activation maps in the cell, because that would be really the stuff that we, and that's a Nature paper.
So what resources would you recommend for students to learn more about this?
I think in my experience, I'm talking to a physics audience here, so it's in my experience, the fear biology is often underestimated. The fear of physics is always out there. People are, physics, and but I saw it among physics students. Biology, they're equally fearful of biology and techniques. So there's, learning the tools. You don't probably need to take a molecular biology course, but you need good training when we have collaborators. So here's the thing. I would say, don't be afraid of stepping outside your comfort zone and learning techniques, whether it comes from chemistry, biochemistry, molecular biology, that actually keep your eyes on the end goal. I'm doing this not because I want to become a God's gift specialist on how to control expression. I do this because I want to achieve this type of expression levels that allows me to do the measurements that I want to do. So if you have that focus in mind, I think everything is possible. I think that's one advice that I would give. Don't be afraid of areas that, I don't like biology. Chemistry or biochemistry is too, it's outside my comfort. If you really want to passionate about the project, I think this should be a sacrifice or a risk that you have to take.
There's another question in the YouTube chat. So
Yeah,
it's asking, could you go over the AOTF in the setup with multiple lasers, and what role does it play in the setup?
Oh, yes. AOTF, acoustic optic tunable filter. So, I can talk about it here. It's basically the same model of AOTF. I can tell first here, and then I'll go to the white light laser. So what it does here, you have a radio frequency that basically, if you change the frequency, you basically can select which wavelength gets diffracted to the first order. And that's basically, and it's a very fast response. So you know that if I want red, I need to apply, say, I don't know what frequency, 150 kilohertz or something. It's you have to do the tuning, and then you have to tune the voltage, the amplitude, to get how much is the percentage of incoming light. So we have calibration curves both for frequency and for amplitude that tells you which wavelength and which amplitude. So you control both the intensity and the wavelength with this. And these are, in this particular case, are 4 discrete diode lasers, but is no different here. What you do, I have an AOTF that's right now it selects 532 and 650, and I'm going to work with an incoming grad student to add 480, so to have 3 color, because we can also want to move to 3 color fret, where you have 3 different labels, but you need to alternate and select this laser. What I didn't mention, you see how they come here, the pulses, they alternate. I have to delay. So if I have pulses, say, separated by 20, say 40 megahertz, then I delay one of them, and I get a 20 megahertz rep rate where one pulse is green, the other one is red. One pulse is green, the other one is, so I need to create delays between these. So IOTF allows me to split the wavelengths, and one goes into a fiber, and then they're recombined. So that's actually, there is quite a bit of optical design and so on to kind of think, how do I modulate my light both spectrally and temporally to do the measurements I do? Because what I didn't tell you in this particular, one does this here, what we could do, it's a pretty cool application that I have to re-educate my grad students. What this you can do in here, you can put a mixture of say something in your sample, and you can do particle sorting. You can say, oh, I have 20% of my particles have a blue label, 30% have only the green label, and the other 50% have both the green and the, so you can do this even if you don't have frets. So this allows you, for instance, to sort particles just the same way that there are setups that sort cells. There is cell flow cytometry experiments where you can actually one cell at a time passes, and you can say high copy, low copy. You can do this at the level of single molecules to do molecular sorting. So it's a pretty cool stuff. It's not only for fret. You can actually do molecular sorting, which is pretty cool. I hope I gave a long answer for a simple question, but and you can also change the polarization of the 2 colors that you change. So you can play a lot of different games here in terms of modulating your incoming excitation light. I didn't talk about the electronics module. There's a very cool electronics module that really tags all these pulses that come through. So,
and the last one from the chat is, what would be the best way for a student to get in contact with you?
Write me an email. I'm not so much on social media, but I do tend to respond to email quite well. I'm an old-fashioned in that mode. So, and if you happen to visit Toronto, I'm always welcoming people to see my lab and talk to my students. So that's something that's always, it's hard for me to say no, particularly to students. I have that. Ben probably knows. Yeah.
Great. I think that's it from the chat. So
Thank you again, Professor Gradinaru, and thank you everyone for participating and attending. So I also wanted to say that if you are also interested in participating to other events from SAC, you can always look up on our website or follow us on our social media. And I also put the link of Professor Gradinaru website on the description of the YouTube live, so you can always go back to his website to if you have more questions or if you're
Yeah,
more
I need to update
interested.
it a little bit. That's
Mhm.
true. I have to do that this summer. There is a bit of outdated information, but I'll get on that. Thank you so much.
So just go back to Professor Gradinaru website once it will get updated. And
Even now, it's
again,
good enough, but I will update it. I have some new information that I should put there.
okay, perfect. And have a wonderful rest of your day. Thank you a
Thank you
lot.
so much.
Thank you so much.
Bye.